Hello, everyone. Good to see you. I'm glad that you're with us today for our environmental health seminar series. I'm Ellen Hahn. I direct the UK Center for Appalachian Research in Environmental Sciences. The people on this call know who I am, and you know all of you are members, which is awesome. We are so excited today to have Dr. Sean, see if I can say it right, Compagna, like lasagna, Compagna. Thank you. And we're not going to hold against him where he's from, of course, but we did have a conversation about the game last night, but we don't have to talk about that. So I do encourage all of you, a reminder, if you are interested and need services through the center, to please use the research portal link that Kara put in the chat, or she'll do it again, I guess. And I'm going to turn it over to Scott Stanley, who is the co-lead for our analytical core services. And Scott will introduce Sean and be the moderator for today. Thanks so much.
Thank you.
Thanks, Ellen. First of all, I want to introduce Dr. Sean Compagna. Very excited to have him here today for his talk. He completed his bachelor's degree in chemistry at North Carolina State University, where he initially did some chemical research on synthesis of bacterial chromosomes. He then moved to Princeton and did his PhD there, working in a collaborative effort to research and characterize the chemical properties of interspecies bacterial signaling molecules. He followed that up by doing a postdoctoral fellowship at the Louis Sanger Institute of Integrative Genomics, also at Princeton, where he developed methods for novel biochemical synthesis pathways of natural products and whole cell extracts. He followed that by moving into a faculty position at University of Tennessee in Knoxville in the chemistry department, where he's been since August of 2007. Today's presentation, as you can see by the title, is probing the microbial metabolism of chloro and fluorochemicals. Reading some of his research, I found that he is doing a lot of metabolomics using mass spectrometry and identifying biomarkers. So I think we'll have a very exciting talk today, and I'll be handling questions after. We'll open the microphone so that you can ask. But welcome, Dr. Compagna.
Thank you for the kind introduction, and thank you for still letting me come after last night. I do appreciate it. But today I'm going to talk about some of the work that we've been doing in not necessarily environmental health, but environmental applied remediation work. And this talk is actually one of my favorites to give because it touches at sort of the core of what I'm interested in looking at bacterial metabolism and bacterial functioning and patterning of the world around us. And so just to introduce myself, I am a chemist through and through who really enjoys biology. So, I grew up in the sort of the beginning of the chemical biology era when that word was starting to pop into vogue. And primarily my lab works on chemical synthesis of biologically active molecules in one form or another. But I was fortunate enough during my postdoc to be involved in sort of the birth of metabolomics in many ways in the Remediation lab. And so we do a lot of bioanalytical chemistry now, primarily high resolution mass spectrometry, with really the hope of understanding how small molecules pattern the world around us. What's really powerful and really great about this technique and really fuels a lot of what we think about is the fact that many parts of metabolism are conserved. And so our platform really allows us to investigate a number of systems beyond just bacterial metabolism and a few other things that I'm personally interested in. And we have a ton of great collaborators, actually from around the world, where our core interacts with them to perform high-level metabolomic services on often a collaborative basis. So that is us in a nutshell. I'm going to start out by really highlighting why an organic chemist got very excited about metabolomics. As many of you know, this is the systematic study of the entire set of small molecules. And in our case, we consider that molecular weight 2000 and under that make up a cell, right? And so by understanding this metabolome, people have been really thinking about for a very long time that we could understand organismal function and physiology in a different, at a much deeper level. And this is actually the oldest of the omics techniques, and I won't go through the history lesson that I normally do, But we think of the birth of omics in our modern consciousness as following the sort of the time that the technologies themselves became available. So genomics and Sanger sequencing came in vogue first. That was quickly followed by proteomics, which was due to a rapid expansion in mass spectrometric capabilities. And the metabolomics followed from that, again, with some new advancements in mass spec technology. But actually in the late 60s and early 70s, Linus Pauling was doing gas chromatography, FID characterization of human urine, and sorry about that, and actually discovered the genetic defect through identifying the small molecules that leads to phenylketonuria. Linus Pauling's efforts were great. It led to the ability to, really treat people with this phenylalanine metabolism issue. But metabolomics, and we'll put it in quotes, at that time didn't catch on because he was routinely measuring about 200 compounds, but only identifying about 10 of those at any point in time. And so the technique was thought to be powerful in specific cases, but the unknowns really scared people. When I started doing metabolomics, in the mid 2000s, in my postdoctoral career, what we quickly realized is we were identifying at that time about 120 compounds. We're now up to identification of several hundred, 400 to 800 compounds, depending on studies. But we're now measuring tens of thousands of molecules that we still don't know what they are. And so this knowledge gap of what we can identify and what is out there still exists. Tools are better. The gap's getting bigger, right? There's more and more things that we're able to detect as mass specs, and NMRs get better to do so. And as an organic chemist and someone who's interested in basic physiology, that's actually a spectacular place to be. It means there's always new molecules to look into and new things to understand. And the other great thing about it is the technology is already great enough or good enough to answer biological questions along the way while cataloging these molecules for later discovery. And so I sort of set up this slide by pointing out that metabolomics is an omics technique that sits in a hierarchy of really great technologies that tell us different things about a system. When we think about genomics, that gives us the information content of the cell. We think about transcriptomics, that really allows us to know what messages the cell is trying to send, regulate itself. And when we look into proteomics, that allows us to measure the structural components and the enzymes of the protein. One of the gaps that is still there in proteomics is that we can't fully characterize post-translational modifications. So oftentimes, even though we can characterize the proteins in the proteome, we can't identify their function, which makes metabolomics, which measures the small molecules, which are the currency of life, a very powerful complementary technique because it can inform the functional physiological side of the proteome and these other technologies. Now, if this was an analytical chemistry talk alone, I would go on for about a half hour or so and talk about the bioinformatic differences between all of these. But I do want to highlight that when we think of genomics, transcriptomics, or proteomics, it's primarily a sequencing effort where we're using different technologies to determine how monomers are organized in a set of macromolecules that are of fairly similar chemical composition. When we go to the metabolome, that's just not the case. There is no code that we have cracked yet that maps the small molecules that we expect back to the proteome or the genome. On top of that, the metabolites are very different in chemical composition. Amino acids are different phosphosugars in both charge and elemental composition. Lipids, again, are very different from any of those water-soluble metabolites. And so the chemical diversity of measuring the metabolome has been a challenge. And as the analytical chemistry part of my lab really loves the fact that there's always new techniques and new ways to get better coverage of the metabolome in general. Now, it's a difficult task, but what's really great about metabolomics is that if you can capture a snapshot of the small molecules in a cell from a system in a well-constructed experiment, you're not only answering the hypothesis that's been posited when the samples were collected, but you're generating a resource that allows people to go back and ask questions from those data over and over again. And so the information from metabolomics can be used at a systems level, where you use the entire collection of molecules through ANOVA and other analyses to ask the question, does the physiological state or collection of molecules indicate that this organism or system is behaving differently than others? And this is a great way to speciate between healthy and unhealthy, or remediated versus unremediated soils, as we'll talk about today. If you go in and look at specific metabolites, you can often map them to pathways and understand how energy and nutrients are being moved through an organism, which gives you insight into how they function at the biochemical level. And then you can even drill down further and look at single enzyme or single metabolite data and understand how a specific enzyme works, or how a specific contaminant or compound is impacting the system. And so for today's talk, we're actually going to start at the single metabolite level, move through the pathway level, and then have a systems level talk at the end. Now, all of this takes a lot of instrumentation to do, and to do well. And just to highlight really the rapid expanses and mass spectrometric technology that have happened over the last 3 decades. When we started in 2005 in the Rabinowitz lab to 2007, thinking about metabolomics, we started on an HPLC triple quadrupole instrument, which actually had to have a standard of the molecule in hand, or parameters that told you how it functioned in the mass spec and in the fragmentation cell to detect compounds. And so my first postdoctoral job was to sit around with another postdoc and just inject compounds into the mass spec and figure out their retention times and fragmentation parameters. And we quickly built a library in about a year or so with about 300 compounds in it. And we could measure about 120 of those just from the water soluble compartment, so no lipids. And that took about 2 hours. Thermo then came out with a mass analyzer called the Orbitrap that popped up in about the same time, which was a Fourier transform high resolution mass spectrometer. I won't go into too many of the details other than to say that it allowed the identification of compounds of the size of metabolites pretty much from exact mass alone, which allowed us to collect data on unknown things, predict their formula, and really expand our coverage of molecules. So we ported our method over to that. And actually on the first run, we detected 150 water-soluble metabolites out of our library of 300, and about 4,000 molecules that we had no idea what they were. And so at that point, the field in general across the US realized that the biochemistry book is probably 1 to 5% of what is happening inside a cell. And there's a lot of molecular structures or spaces that are very important for life on this planet that we don't quite understand. So now we've evolved past that. And in the core here, which is run by the Associate Director, Hector Castro, we actually have a suite of instruments. Not all of them are shown, but we routinely run an analysis for water-soluble metabolites, where we quantitate and measure about 150 known compounds and about 8,000 spectral features from unknowns. We often run a lipidomics analysis where we detect about 400 known lipid-like compounds from our library and add information from about 12,000 other spectral features. And then we have instruments to do boutique analyses for targeted sets of compounds, for instance, like the cobamides, like you'll see today, prostaglandins, and so on and so forth. But the reason I popped this up was just to impress upon everyone that in about an hour of instrument time, or real time in about an hour and a half of instrument time, we can now get quantitative information on about 20 to 30,000 molecules from a biological sample. The work of Dorenstein out at UCSD and Scripps to build the GNPS platform, which allows global annotation of metabolites through community efforts, is just making these tools and techniques more and more powerful, with more and more people being able to interface and interrogate the data. So that's the technology platform that we work on, and I'll be glad to answer more in-depth questions or provide more details after that, or after the talk. So now I'm going to shift to discussing the bioremediation efforts that we've been working on with Frank Loeffler's lab and the Center for Environmental Biotechnology here, and tell you hopefully 3 stories, time permitting, about work we've been doing to understand how microbes can be used to dechlorinate and potentially defluorinate common contaminants that are found in Superfund sites, military bases, and other industrial places around the world. So the first talk we were going to discuss, I told you, was going to be at the metabolite level. And so actually this was, I think, the first project that Frank and I did together. So he came to me. I was interested in Desulfidobacterium hafniase. And this organism is able to dechlorinate a variety of compounds. It's a gram-positive organohalide respiring organism that's important in bioremediation. What was interesting with this to Frank is that they knew that this was a cobamide producing organism, and that these cobamides, which are analogs of B12, are used in many of the reductive chlorinases that are employed by these bacteria to dechlorinate these chemicals. And so here you can see on the right, that is the structure of B12. But to date, there have been a number of compounds that have been found in bacteria where the entire structure is conserved, except for this bottom portion here that is called the lower base of the lower ligand, depending on whether or not there's nitrogen to coordinate to the cobalt or not. And you can see that the range of lower bases and lower ligands consist of fairly complex heterocycles all the way down to relatively non-complex phenols. What's intriguing about this is biochemically and the way it works in the soil. Not all microorganisms make the cobamides, some do, some don't. So there are mechanisms for exchange of cobamides, removal and replacement of lower bases, and so on and so forth. And it's becoming clear that just by the structural modification in this lower portion of the molecule, that microorganisms are tuning the way enzymes function based on how the cofactor fits with just lower base modifications. So Frank's lab came to us after doing some HPLC UV traces of the cobamides, which are very brightly red colored, and noticed that in the dihafniase strain, that the cobamide they were detecting had a different retention time than any of the cobamide standards that they had previously employed and collected. So this wasn't vitamin B12, it wasn't the phenolic version, it wasn't any of the known standards. And so then they came to us and just asked the simple question, can we help characterize the structure of this molecule? So my grad student, Alan Borden, first off ran the mass spec and determined that the cobamide from dihafniase had a mass of 1329.54. Vitamin B12 has a mass of 1355.58. And then if you think about only the lower portion of this molecule being different, you can actually do a mathematical subtraction between the mass of the conserved portions of vitamin B12 by removing the dimethylbenzimidazole portion. That tells us that the conserved portion of these molecules, I guess I don't have the exact number and I'm not going to do the math in my head, but if you take the mass of the unknown cobamide minus the mass of B12 minus what we know is the non-conserved portion here, this is the total conserved portion of the molecule. That told us that the lower base or lower ligand in this case weighed 120.04 Daltons. Now, if you use this high resolution mass spec data to determine structure, you got one in exactly one hit, which was telling us that this lower base was C5H4N4. And when you think about naturally occurring heterocycles, the only one that fits that structure is purine. Now, we were pretty certain at this point that we had found a purinyl cobamide, but mass spec isn't actually a structural technique. Instead, it just told us that this was highly likely based on biological information, but it doesn't actually tell us anything about the structure. So Alan used actually a couple of really interesting manipulations of the molecule. First, to make sure that there were 4 nitrogens involved and not some other elements. This needed to be done because our search is actually constrained elemental composition, and we wanted to make sure we didn't miss anything. So Alan then grew the dihafniase strain in heavy labeled nitrogen 15 instead of normal nitrogen 14 ammonium chloride. When you do that, all of the nitrogens of the molecule weigh 1 more than they used to. This allows us to then count the number of nitrogens in the mass spectrometer by going through and just looking at these mass shifts. So in our case, we knew that the parent molecule in this case was 1329 for our hafniase strain. And we noted that there was a mass shift of, I think it's 14 or 15 here. When you subtract the conserved nitrogens, this conclusively showed that our lower base contained 4 nitrogens. So now we were 100% sure that our chemical formula was correct, and we just needed to know if the connectivity of the molecules was also correct. From there, we turned to NMR, or nuclear magnetic resonance technology. And we characterized both vitamin B12 and then our cobamide here. As you can see, vitamin B12 is a trace in blue. The cobamide is a trace in black. If you remember back to your organic chemistry days, for many people, NMR is a technology that allows us to count the number of protons in the system. It also allows us to know whether or not the protons are connected to a carbon that also has a proton based on the amount of splitting. So all of these signals down here in the bottom of the molecule come from the conserved ring portion of both of them. And so we can discount those. However, these aromatic protons in what we thought was purine or in the benzimidazole base here on vitamin B12 sit in a fairly privileged portion of the molecule where they're here outset. I won't go through many of the details, but you can notice that none of the protons in B12 are connected to a carbon that has any other proton. So there's one isolated proton here, an isolated proton here, and none of the neighbors have protons. And so they should all be singlets or single peaks. And you can count those out in vitamin B12. When we went to our structure, again, all of the peaks were singlets. One of the only ways to get that connectivity from the singlets was to actually have purine. Alan went through and through some two-dimensional NMR that we won't talk about today. He assigned the fact that we had these 3 singlets to these 3 signals here. And this, for the most part, structurally confirmed that we had purine in this cobamide. Now, once we had determined the structure of this cobamide, which was, I think, the first structurally characterized cobamide since 1993, dates may be wrong there, but definitely in over 2 decades, we wanted to ask the question, does it have different biological function? And how would this fit into an ecosystem? And so the first thing we asked was, does the purinyl cobamide support chlorinoid-dependent reductive chlorination? And if you look at Dichlorobacter restrictive strain, which is very close to the Dulcifidobacter strain that we were studying, you'll notice that if you add vitamin B12, you quickly see perchloroethane or PCE disappear, and you see trichloroethane start to appear and then quickly go away, and cis-DCE or cis-dichloroethane become the primary molecule relatively quickly. If you add pure purinyl cobamide, again, you see the same behavior. You see a very rapid drop off of the perchloroethane, followed by production of cis-dichloroethane. And if you don't add chlorinoid, you get no activity. And so we were fairly certain that this closely related RDase enzyme, which had a 98% match with the one from our organism, structurally was able to take this purinyl cobamide and function. Now, if you looked at the Helicoccoidus mccartyi, which has an RDase without much of a match, less than 20%, but you can see that it is highly active in dechlorinating cis-dichloroethene to vinyl chloride. If you add vitamin B12, the ultimate production of ethene. If you add purinyl cobamide, there is activity, but it gets stuck at vinyl chloride and the activity is very, very slow. And if you don't add any chlorinoid, you can actually just see some probably leftover degradation from what the cofactor was there before. And so we were fairly certain at this point that we not only had discovered a novel cobamide, but that this novel cobamide expands sort of the repertoire of lower base structures that can modulate chlorinoid-dependent enzyme function in these organisms that are very important for the remediation of chlorinated chemicals, both environmentally and commercially. So from there, we actually started looking at another set of organisms. that are from the Peptococcaceae family, and trying to understand how they dechlorinated dichloromethane degradation. And so here's where we're going to get into sort of the hardcore biochemistry, multi-molecule pathway analysis of things. And dichloromethane is actually a major contaminant. We produce 260 million pounds of this yearly. And in the US, dichloromethane is an $850 million industry that was projected to hit 1 billion in 2019. I don't have numbers on how the pandemic impacted this. This is a major paint stripper, degreaser. It's used in plastic glue if you like to build plastic model kits. It's used to extract hops from beer. It's actually used to decaffeinate coffee. And so all of your decaffeinated coffee has had a dichloromethane extraction along the way. The problem with this and many chlorinated chemicals is they have a density that's heavier than water. So they end up sitting on the bottom of groundwater systems, which means that oftentimes, aerobic and anaerobic degradation degradation can be difficult because of where they sit, even though it does occur. And so degradation needs to happen before they end up in the water table. So in this system, actually the initial hypothesis of this study, or an initial goal, was again to characterize a single molecule. And so what we'd become interested in is how the Wood-Lundahl pathway, which is a pathway that takes single carbon molecules and puts them into folate metabolism, was functioning. And so the canonical Wood-Lundahl pathway starts with methane and ends up making a couple equivalents of CO2 and an equivalent of acetate through the metabolism of methane. In this case, we had hypothesized that chlorinated chemicals like dichloromethane were going to enter a Wood-Lundahl pathway. All the genes were there, and we were really trying to ask the question, exactly how did that happen? Was it through full dechlorination through some unknown enzyme to methane, and then methane just directly entered the Wood-Lundahl pathway? Or were these organisms using modified folate metabolism where they were generating chlorinated folates along the way? Frank and I were actually really hoping for the chlorinated folates pathways because it would have been incredibly novel. And I can tell you we have not as of yet detected them or answered that question. But what we found along the way was actually fairly interesting. And so for these studies, we looked at a Halobacterium formicoaceticum, and I do not know why my Latin is so terrible. I do apologize. And so they were the first known bacterium to be able to degrade dichloromethane, and they were isolated in 1996. Since then, a Candidatus species that must work in a consortium of other organisms has been discovered that has this dechlorinating activity on its own. Now, no one knows why we can't isolate the Candidatus, even though this study, I think, will maybe give some information on that along the way. And what we wanted to understand is whether or not a consortium versus an isolated species were using different Wood-Lundahl pathways, and also hoping to find these molecules. So from there, we actually performed an experiment using carbon-13 labeled, again, a stable isotope labeled dichloromethane, where we took live cultures of either the consortium or the isolate, grew them in an NMR tube, and just left them in the instrument, measuring these small molecules over time. And so what we found in the isolate was that very quickly, if you looked at the carbon-13 NMR, which gives a good signal based on the carbon-13 dichloromethane, we detected glycine, acetate, methanol, and carbonate incorporating the label from the stable isotope labeled material, meaning they were being produced through metabolism of this organism. If you look at the proton NMR, we detected acetate, methanol, glycine, and saw almost complete consumption of dichloromethane. Previously, I told you that you should see a singlet if the carbons are by themselves. However, the carbon-13 splits them, so the fact that you're seeing a triplet for acetate, methanol, and glycine also further confirms that carbon-13 from the added dichloromethane is making it into these molecules. From there, we moved to looking at the RM consortium, and what we immediately saw was we saw almost no acetate, very small amounts of methanol, but we saw a lot of carbon monoxide, a lot of carbonate, and a lot of carbon dioxide. So we were seeing a lot of mineralization, but we weren't seeing much fermentation at that point. Further, if we looked at the proton NMR, you'll notice that we did detect methane, acetate, and a very tiny bit of methanol. However, our methane and acetate are singlets, meaning that they were not being produced from whatever pathway was degrading dichloromethane. From there, we started to think about exactly what that means, right? First of all, it means that we would have had to be using two different parts of the Wood-Lundahl pathway. The organism candidatus from the RM consortium wasn't producing acetate, so it wasn't using all of the Wood-Lundahl pathway, to where the isolate it definitely was. So Frank's lab did some thermodynamic calculations, and they noted that when you're going to ferment DCM through the equation shown here, that the fermentation of DCM does not depend on the concentration of hydrogen in the system. However, if you think about DCM mineralization is very favorable, more favorable than fermentation at low hydrogen concentrations, and much less favorable at high hydrogen concentrations. The energy goes up for that pathway. So that actually sort of informed why the consortium is so important, because we knew that two of the organisms in the consortium, one was an acetate producing bacterium, and another one was a CO2 methane producing bacterium, both of which reduce the partial pressure of hydrogen to produce methane and acetate in this culture. So what we now think is that the consortium only uses one half of the Wood-Lundahl pathway to perform mineralization and not fermentation using this pathway, by taking in dichloromethane and going to CO2, while allowing the offshoots of the Wood-Lundahl pathway to be used by other organisms in the consortium, to where the isolate is using both parts of the Wood-Lundahl pathway, primarily to do dichloromethane fermentation. And so this highlights how looking at a systems level approach through NMR metabolomics allowed us to differentiate pathway utilization, even though the genes of the pathways were highly similar between both of these organisms. So now for the last vignette that I'm going to pop up, we're going to again talk about dechlorination. In this place, or this time, talk about reductive chlorination. I previewed this earlier when we were talking about the cobamides. We talked about taking perchloroethane, where each of these red atoms is a chlorine and the blacks are a carbon, and that there are organisms that could sequentially take this to ethene, which is non-toxic, and all the while producing all the energy necessarily to run their metabolism from this. This is called organohalide respiration, as we noted before. Now, one of the problems with using this process of organohalide respiration for bioremediation is that as perchloroethane moves through the water supply and is transformed to trichloroethane all the way down to ethene, you have some relatively toxic intermediates when you have 2 to 4 chlorines involved. However, when you go from cis dichloroethane to vinyl chloride with only 1 chlorine, you actually make the most toxic intermediate in the entire pathway. And then ethene is entirely benign. And so if something happens where you're not efficiently converting your heavily chlorinated molecules to vinyl chloride, or you're stopping at this point, you actually can have the opposite effect. Instead of cleaning up the environment, of making even a more toxic compound and a bigger mess. This is further made difficult because the consortium SDC9 that is used actually has a number of organisms that take the fully chlorinated compound to cis DCE, but only a very few organisms in the dihalocoides that take the cis DCE all the way to ethene. And so there's a propensity for stall at this region of the chlorination process. And this is even highlighted further because many of the biological inputs and metabolites that dihalocoides needs to survive, proliferate, are produced by the consortium during the beginning parts of the dechlorination. And so if one looks at how this consortium functions, either in the presence of dichloroethane or vinyl chloride, you can see that you stimulate growth of the consortium with lactate in the presence of the chlorinated energy sources. And you can see that the total bacterial copies quickly spike and then drop off. And then really late in the growth phase, the dihalococoides sort of take over and complete the dechlorination process. This happens for both. If you start with vinyl chloride, you can see the dihalococoides aren't quite as dependent on the spike of the total bacteria, but again, they only come about later once the lactate has been almost fully removed from the culture. So there's some preconditioning of the growth medium that needs to happen before the dihalococoides take off. So the goal of this experiment was to go in and actually ask the question, could we use metabolomics to determine where we were in the process of dechlorination, potentially learn something about the process along the way, but primarily use this as a way to go out into the field and inform people who are doing bioremediation efforts as to the extent of dechlorination, and also to allow them to know whether their SDC9 consortium that had been injected were still functioning. So we performed 2 experiments where we took time points along the dechlorination process from T0 to T5 over about 50 days or so, and T0 to T5 here again over about 50 days for both vinyl chloride and cis-DCE. This just shows how the ratio of the chlorinated inputs changed in our experiment. You can quickly see that cis-DCE disappeared. Vinyl chloride was produced in the first couple of days or so, where the organisms are living off the mixture of chlorinated inputs, and then ethene eventually was the only molecule that could be detected. Vinyl chloride looks a little simpler because there's only one stage of chlorination, and you can see that as vinyl chloride disappeared, ethene was produced. So when you look at the metabolomics experiment, we're not going to talk about any specific molecules in this and just talk about it at the systems level. You can see that each of the time points, we measured between 7,000 and 9,000 molecules that were indicative of the physiology of the system. Again, I'll highlight, we only know what about 200 of those are, and we haven't measured lipids at all in this case. There's still a huge knowledge gap, but even with that, using partial least squares discriminant analysis, we were able to note that the time points clustered in different space. And so if you look at time points where dechlorination either hasn't started with the black dot shown here, or dechlorination was fully over with the red dots, they look very similar. There was no dechlorination activity that was indicated by the physiology of these organisms. If you look at the points where we're living off a mixture of vinyl chlorinated compounds at T0 and T1, it's still primarily shifted towards cis-DCE. You can see that those points are distinct. And then as you move later and start to run out of cis-DE and chlorinated compounds in general, that the metabolism of sort of these middle points where we're finishing off our dechlorination process look very similar before, like I said, the final return to a physiological state that looks much like the initial state. Quite frankly, I think all of us were shocked that we got this much separation on these, and we're able to distinctly tell these apart, especially with the number of unknown features. The vinyl chloride story looks much the same, and I didn't put in animations for that. But again, you can see that early and late time points cluster together to where the time points in the middle sort of go through and match with the amount of vinyl chloride that's left behind. I left out the next slide on our field studies because they're not quite complete yet. The pandemic has made it hard to find things out or find sampling opportunities in the environment. But we're currently working with a number of people to go in and ground truth these, hopefully find about 20 of these molecules, either features that we already know what they are or unknown features that can be used for targeted studies to go in and really monitor remediation at a number of sites. So I lied to you a couple minutes ago when I said this was the last vignette, but in the last 4 minutes or so, I will hold true to the title and talk a little bit about fluorochemicals. Now, we all know that PFASs in general are becoming a major point of concern, and chlorinated fluorochemicals were concerns long before that. And so now where we're shifting our efforts are to find an organism that can help in remediation of fluorinated chemicals. To date, we haven't found anything that can directly break the carbon-fluorine bond, which is a much, much stronger bond than carbon-chlorine. And frankly, it's debatable that we'll ever be able to find that. I hope so, but biologically, the thermodynamic calculations don't quite make sense yet. However, the Loeffler lab did find a Pseudomonas species strain 273 that could use both chlorinated alkanes as a sole carbon and energy source. And see, here you can see it surviving off 110 dichlorodecane, and ran a study with it and asked the question, will it grow on fluorodecane or 110 difluorodecane? And the answer was yes. Now, we don't think that this, like I said, is through direct breakage of the carbon-fluorine bond. Instead, what we think is happening is an oxidation of the fluorinated carbon, which then leads to spontaneous loss of the fluoride, followed by oxidation of that aldehyde to carboxylic acid. So we were studying this metabolism in general just to look into enzymes that could attack the non-fluorinated positions on PFASs. And we actually found something fairly unique when we looked at the lipidome and we're trying to ask where these inputs were going and whether or not there were changes in the lipidome. What we found is that on 110 difluorodecane, you have a fluorine at both ends. If you think about oxidation of one of those carbons and then going through normal beta oxidation to make 2 carbon inputs, you should have a fluoride left. And we were really wondering how that fluoroacetate, which is known to be a metabolic poison, would be tolerated by the organism. What we found was actually fairly intriguing was that almost 90% of all of the lipids had incorporated fluorine into the lipid through the incorporation probably of monofluoroacetate. We haven't fully mapped out how that metabolism works quite yet, but what we do know is that using mass spectrometric techniques to look at fragments, that we are finding monofluoro inputs into a large number of lipids. And if you look here on cultures that are grown with either decane, decanoate, 1-fluorodecane, or difluorodecane, you can see that we see peaks shown here in the solid lines for phosphatidyl and ethanolamine here that has 34 carbons and 1 double bond in the tails. In decane, decanoate, and fluorodecane. But when you look at the difluorodecane, we primarily see that lipid as having 1 fluorine in it. We've also confirmed this by looking at the fatty acid tails, and you can see again here that in the difluorodecane cultures, and to some extent in the fluorodecane cultures, we're seeing C16 and C18 fatty acids that are containing fluorines. This is actually a really interesting potential sink for fluorine and fluorinated compounds in the environment that was relatively unexpected. It's also a really interesting biochemical story. There have been organisms that have been shown to make fluorinated fatty acids. There have been organisms that have been shown to metabolize fluorinated fatty acids for inputs into metabolites, but we haven't yet seen an organism that can take fluorochemicals, produce the fatty acids, metabolize them, and then resynthesize new fatty acids and then incorporate them into lipids and other macromolecules. And so we're very interested in this organism. We're very interested in how it's going to interact with PFASs and PFOAs, either the entirely fluorinated ones or the polyfluorinated ones as we move forward. And so with that, I would like to thank the people who were involved in this study. On the right, you can see my group members here. Dr. Amanda May works in the Center for Environmental Biotechnology and was the lead is the current person that's the lead for my lab on many of these studies. Alex Walls here didn't get bolded like he should have. He's just joined the project and is working on it as well. Much of the work you saw today was done in my lab by Abigail Tester, Alan Gordon, Alex Fish. This is the group of collaborators, Frank Loeffler, Gao Chen, Jun Yan, and Yangqi Zi from the Center of Environmental Biotechnology here. We also partnered with Dora and Sam from Microbial Insights, a local company right down the street for much of this work. And then Mandy Michelson from the Army Corps of Engineers also was very instrumental in helping us find some startup funding to do many of the early studies as well. With that, I thank you for your attention, and we'll be glad to answer any questions that you have.
Thank you, Dr. Compagna, for that exciting talk. Found it very interesting how you incorporated so many of those different elements. I'm going to ask that anybody that has any questions that they haven't put in the chat to jump in, but I do have a couple first that I can ask from Dr. Mosley. Put forward a couple of questions for you. And the first one Dr. Mosley put forward was for the DCM portion of your talk. He said, or he asked, could a doubly labeled C13 deuterium DCM provide a more specific information for the enzymatic steps?
Possibly. I'm trying to think about that. Yeah, I don't know that the deuterium would help much. And hi, Hunter, it's good to see you again. It's been a couple of years. So.
Yeah, it has. I know. It can't go to conference. We haven't been able to go to conferences like we used to.
Yeah, we definitely need to chat some more. So I think those steps are pretty worked out as far as the dechlorination step goes, Hunter, because they don't use the ethene for anything. They just use the chemical potential from breaking the carbon-chlorine bond. And it's actually a direct breaking of the carbon-chlorine bond. The carbon-hydrogen bonds stay in place for the entire time. So I think for the studies with DCM, it would be far more interesting because we don't know if that's, I mean, it's probably direct breaking of the carbon-chlorine bond, but we don't know how those protons interact. So yeah, I need to think about that. There should be a kinetic isotope effect at least in one of those steps, right?
Well, sometimes it shows you there's more than one enzymatic mechanism that's utilized by different organisms for the same steps. So it can help separate, give you specificity on the enzymatic mechanism, because sometimes more complex things, they pop the hydrogen off to create carbanion that's then it goes through other steps to get to your ultimate goal. But it's amazing just how complex some of the enzymatic steps are.
Yeah, no, that's actually a really good thought. I'm gonna have to, I think you just got Alex some work to do for the next couple of months. So.
Hunter, I'm gonna let you go ahead and ask your other question then, since you're on.
So it was interesting. Very nice presentation, by the way, Sean. And by the way, that the fact that the biofilms, conditions have to change over them, where you showed the lactate removal before, because the lactate's probably out competing somehow it's out competing or changing enzymatic activity. Could you knock out, figure out what's the lactate transporter on the bacterial strain performing the step, knock it out, see if it's viable, but that might then optimize the biofilm to do all the steps more quickly.
Yeah. So Hunter, I think the more important, I think the loss of lactate is actually an artifact, right? That's what you're getting to get the other bacteria to grow. I think it's actually the removal of H2, hydrogen gas, that's more important, and the lactate is just a stimulator to build the associated microbial population to a point where their metabolism can pull the H2 concentrations down enough, right?
So it's more of a biomarker of the process, not a direct.
Right.
Understand.
Yeah, that's our current thinking, right? So correlated and not causal.
So I have another question online from someone. Dr. Lee is asking a question, and his group has an interest in lung-induced injury from inhaled chlorinated compounds. His lab was working with Dr. Morris last year to measure the chlorinated lipids in rat plasma after exposure to chlorinated gas inhalation. The data was a bit inconclusive, and he was wondering if it's possible to use the bronchoalveolar lavage fluid that may be a better measurement for those chlorinated, I assume for the chlorinated compounds.
I mean, yes, potentially.
Sorry to interrupt you. Actually, was chlorinated fat lipid acid, yeah, fat acid. So it wasn't totally statistically conclusive. So Dr. Morris left here, as you know, is no longer available for the service, but we're still looking into it. Hopefully be able to repeat it sometime later.
Yeah, so for us, being an analytical chemist, the more places you look, the better off you are trying to find these reservoirs of molecules, right? And I can tell you that oftentimes tissues and fluids will have vastly different concentrations of things.
Okay. Yeah. All right, thank you.
So is this Cl2 addition across the double bonds on these? Like what's the mechanism of adding Cl2? Do you know?
Probably in the chat did not write clearly. We exposed the rats. We're interested. I'm a pulmonary physiologist by training. We're interested in how the chlorine gas can cause lung injuries through the neural mechanism, neurogenic inflammation and edema. So we expose the rats to chlorine gas mixtures and then take the plasma sample to analyze how much chlorine actually get into the system, generate the systemic physiological change in addition to lung injury. So yeah, that's where we took the sample and hopefully be able to find another set of endpoint. Wasn't clearly indicated from our results.
Are you just interested in the total amount of chlorination? Or
yeah, that's right, exactly. Yeah. So it's almost like we have demonstrated the lung injury and also demonstrate electrophysiological response from the nerve ending innervating the airways. But we would like to be able to find a biomarker, if you will, be able to compare our exposure to other laboratory using the chlorinated lipid as a biomarker. That was
yeah, I think if you just want bulk chlorination, then probably induction coupled plasma optical emission spectroscopy or ICP mass spec is probably better for total, because this is going to, I can imagine this is going to be a ton of different lipid species, right?
Right. Yeah. So, yeah, good suggestions.
Yeah. Do you think you see chlorinated prostaglandin type molecules? Will this cyclize arachidonic acid chemically?
We haven't done that. No, haven't even thought about it actually. Yeah, at the time we did the experiment. Yeah.
Right.
Well, I don't believe I have any more questions unless anyone else participating has anything. Dr. Campagna, thank you so much today for your time and preparing this information for our group. It was very informative.
Right. Thank you. And Hunter, we need to get in touch. We talked research pre-pandemic. We need to get back to chatting. So
absolutely.
Yeah, thank you very much.
That's great. Thank you, Sean. This was great. And I would encourage, Sean, we may think about connecting Luan Lee with you to have further conversations, Luan, if you're interested. But we can chat about that. We really appreciate your time today.
Yeah. And just let everyone know, we have a full fee-for-service metabolomics score here that's relatively inexpensive compared to others for a lot of weird state budgetary reasons. So anyone who needs services that aren't offered at UK, we're glad to help. We love to collaborate. it helps us build technology and friends and do great science. So we'd love to be more connected to the community.
Thank you, Sean.
That's awesome.
Yeah, great.
Thank
Thanks
you.
everyone.
Yeah.
Thanks everyone. Appreciate it. Have a good day. Thanks, Sean.
Bye.